In this demonstration we will look at the Hotelling t-square test and at Manova. So as usual we set the working directory in the beginning. You should be familiar with that now. And then we load some data that has been stored on the server. It's a Tibet skull data where different skulls have been found in areas, and the question is, are these different types of skulls similar? So are these different types of skulls, or do they originate from the same population? So you see a couple of variables that describe these skulls. And type 1 are the ones that have been found in graves in Sikkim and neighboring areas of Tibet, and group 2 are skulls that have been found on the battlefield. And so the hypothesis has been that the Tibetans might be survivors of a particular fundamental human type and unrelated to Mongolian and Indian types that surrounded them, and you could check that with these skulls whether they were related to those that have been found on the battlefield. Anyway, that's not our main interest, whether this hypothesis is wrong or right. We mainly focus here on how we do the testing. So we check also that all with the structure command that function, that all of the variables are in the correct form, so as numeric and integer in terms of type. So type should actually be a factor, but it's automatically done if we run the hypothesis testing. And then we extract these data, the first 5 columns, or for the skull data set. That's mainly done because to make things easier later on. So the first thing we need to do is to check the model assumptions for the Hotelling t-square test, and we do this with the MV outlier package. We use the chi-square plot. You've already encountered this type of plot in the previous session, so we don't go into detail, and you know that for low sample sizes we can't expect a very close relationship between the ordered robust Manova distances and the quantiles of the chi-square distribution, but we see that there's overall a satisfying trend, so that's sufficient for our purpose. There's no strong deviation from the multivariate normal distribution, and the same holds for the second group of skulls. We now check the assumption of the homogeneity within group covariance matrices. This has been discussed in the lecture. To check this, we use a hypothesis test. This hypothesis test is a permutation-based test and relatively resistant to outliers and to deviations from normality. So we first use the distance. We first create a distance matrix that's built here on the dist function on the Euclidean distance. And then we use this better dispersion function if you want to know more about how this measurement is done. The dispersion is measured, then have a look into the function and the related paper. We just use the beta disper function now and have as one argument our data, and as second the grouping variable, which is the type of skulls. So we execute that, and we obtain besides some eigenvalues. So this multivariate dispersion is again based on some eigenvalue methods some principal coordinate axis you see here, and we have an average distance to the median of the data. So we now run a permutation test. We used the set seed command before, so we all obtain the same results. And we see that there is there is not much difference. We already saw that the average distance to median is very similar, and we see here that the differences are not statistically significant. If we checked visually for homoscedasticity of the covariances, I could do this with the library PCA-PP, and we plot the covariances of the two groups. Let's look at the results. So that's nicely visualized here. On the lower triangle, you have the absolute values of the covariances, so it's not correlations, it's the covariances. Along the diagonal, makes not much sense to display, obviously. That would be the variances. And here we have, for the individual variables, we have the relationships, and we see, first of all, we see that most of the circles are relatively round. That means that there is no correlation or only slight, so no close relationship between two variables. Only if they become elongated and very long and elliptic, then we have a close relationship. We will see this for another data set. And we see that the circles match each other relatively well, so there is no need to assume, based on the graphical checking as well, there's no need to assume that we have differences between the two data sets. And in addition, there's no indication here that we have a strong collinearity between variables. So we can proceed. If we want to look at the variances and compare them, we can do this as we have done before for a normal ANOVA. You might be aware of this code from the previous code on ANOVA. So we just plot here the two groups. These are all individual observations from the two groups. With such few observations we can do this, and we see that the scatter and the range of the data is relatively similar, so there is no need to, so we don't assume for this variable that there is, so length, for the length variable, that there is a difference in the variance. And you could do this repeatedly for all other types of data, for all other variables as well. So now we can do the Hotelling t-square test that's contained in the library ICSNP. We load this library and then we execute just this. We just execute this function. It's called Hotellings t2, and you just enter your data set that contains the response variables in matrix form, so it doesn't accept it as data frame, and you have as an explanatory variable in this formula interface, you have the two types of groups. If we do this, we obtain a p-value, so we obtain a t-square value, gives you the degrees of freedom and so on, and the p-value, and we see that there is a statistical significantly, that the two groups are statistically significantly different. We could also use the Manova function. This is shown here. We will use the Manova function later on as well, and use the Hotelling-Lawley test type. As we have discussed, there are different test types for the Manova for the checking, and we see that this is also statistically significant. In fact, the p-values are exactly the same because the t-square values are also the same. In case that you have data, we have a strong deviation from the multivariate normal distribution, an alternative non-parametric test could be the grammar test. find here the literature information as well if you want to look. However, this test also comes to the conclusion that there is a significant difference between the two data. We now turn to the Manova. For the Manova, we use data from soils, from the study of Holmgren et al. For us, it's not that interesting what the study was about. It was basically about interactions that facilitate woody encroachment in boreal peatlands. So to use this data, you need to follow this link, download the file, it's table 1 environmental and so on. Simplify the file name to table1 underscore dot xls and move this file into your working directory that you have set for R. Then you can read the file using your path. You can also, of course, put the file in any other directory and use the respective path. And we use the XLSX package to read this Excel file. Read the file. We have a look at the top of the file, and what we see is that this file has several columns. Many columns are actually non-defined, and we have several values that are not defined as well. So they are empty. We see that here as well. We have many NAs in the data set, so we need to remove them before we work on that. As a level, we take from the soil data, we take Hammock. So Hammock is the factor we will use. It's not necessarily that it makes the most sense scientifically, but we are just using this data for demonstration purpose. We restrict the data to the levels A, B, C, and D. That's what we do here. So we extract the data, we check the data, and we now see that we have still a couple of columns left, but our data, our factor is only related to these different data sets. So we remove the non-needed columns and rows, and in addition, you can check that yourself, which columns are removed and which ones are kept. You will see this, that we basically only keep information on the concentrations of nitrogen, phosphorus, kalium, and so on. And we rename the column names to make it easier, and then we remove the non-defined values. So if we now look into our data set, soil dat 4, we see that now we have complete data for all columns, and we have first column that provides us with different grouping information. We extract the quantitative data in a different data frame, and we first check the assumption of multivariate normality. We have discussed before that for small sample sizes, and the sample size of these data sets are relatively small, about 20 observations, we can't expect a really smooth chi-square plot or matching with the chi-square plot of our Malobis distance, but we see that overall it matches reasonably with the data. The same holds here. We see it approximates, it's somehow scattered along the one-to-one line. Same for the group C. For the group D, we see that we have a relatively strong deviation because of one outlier. If we remove this, so by clicking the left mouse button on the point, you remove the point, we can see that now if we remove two outliers, our data fits the multivariate normality criterion much better. With the right click of the mouse, we obtain the observation numbers that are outliers, and now we want to remove those. So we check first what is the first and the 13th observation, what is the case number, and look at the row numbers. The first is 31, the 13th is 113, so we know that these are the row numbers related to these cases. And then we remove these row numbers from both data sets. We have done this a couple of times before in the course. Should be familiar with removing data from data sets. Now we check again. Still not perfect, but much better fit than we had obtained before. Now we check the assumption of the homogeneity within group covariance matrices. So we first calculate the distance matrix. If no additional argument is provided, this is the Euclidean distance that is computed here. And then we run the beta disper function. You need to provide the distance matrix for a data set and the categories. So what we obtain here is we obtain the number of positive eigenvalues. This is always the number of equals always the number of variables in the data set, and we obtain the average distance to the median for each individual group, and we see that group A and B have a larger distance or a larger spread of the values than group C and D, which are relatively homogeneous. So we now do the permutation test. What we obtain is here we have statistically significant differences in the dispersion between groups. So we see that this difference in the dispersion, especially of C and D compared to A and B, is statistically higher, significantly higher, and we need to consider this in interpretation. So we shouldn't use a test statistic that is susceptible to violations of these assumptions. Now let's have a look at collinearity. We see here that for N and K and PK, basically the covariance is from both groups. Shows no relationship, whereas N and P of group B have have a higher correlation. Some correlation is present for these data set, and we see that for group A and D the pattern is the same for Kn and Kp. No obvious pattern covariance present, whereas N and P show some correlation, some association. So we can conclude that despite the statistical significant differences, the overall pattern is relatively similar across the different groups. Now let's run the Manova. That's simply done with the Manova command. You need to take into account that the input file should be a matrix for the response data. So you need to transform this into a matrix, and it can be explained by this grouping vector. We look at the results. We obtain the results for pillarized trace, which is the default test statistics, and there's no statistically significant difference between the different groups of soil. The Wilks test statistics yields to the same conclusion, whereas if we use Roy, we actually find the statistical significance, and keeping in mind or remembering from the lecture that Roy is most susceptible to deviations from homogeneity of the covariance matrices, then we can conclude here that the result for Roy is not reliable. Since we have also a very fairly balanced design, we should the Pillai test statistics should be relatively minor. There should be only minor influence of this deviation from the assumption on the test statistics Pillai. Using Pillai, we can also calculate the average R square. It's just here for additional information. That's not crucial for most of the users. And we can call the summary AOV. That gives you the results for single ANOVAs. And we see that if we would have tested individually the variables, that for nitrogen we would see a statistical significance difference, whereas for phosphorus and sodium and potassium we don't see that. Note that we have multiple tests here, so we should generally we would need to correct, and then the result probably wouldn't be significant for nitrogen either. We have also a non-parametric Manova test, not the permutational Manova, but here the rank test. If you run that, it yields to the same result as for the normal Manova, and also indicates that there's no statistical significant difference between the different groups. We can also run a Manova using the RDA. We have discussed this in the lecture briefly. This is done here. We can just use the data set and explain that by this vector and use the RDA function. And then summary RDA provides us with B-plot scores for the medians of the groups and scores for the sites. We also obtain the information on the overall explained constraint variance by this grouping factor, and that's just 6% in total, so that's not really much. Then we run the ANOVA command to see whether we have a statistical significant, whether the model is statistically significant, and we see that this is not the case. So the p-value is 0.11. We don't have statistical significance here either. And finally, comparing this, so we see that the p-value is actually a little bit lower of the permutation test, but still not significant. RDA offers also the possibility to plot the result, and that's what we do here. First we plot the variables. We use scaling 3, so we see that when we have the first RDA axis, we see that potassium does contribute least to differences, so shows more or less no variation between groups, whereas P and N exhibit some variation across the groups. Then we add the grouping variables and we see that the media, so the centers of the individual groups, they are all very close to the origin of the RDA axis, which means that there is barely a difference between the group medians. And then we extract the scores from the RDA and plot the individual sites in the related cover. And here we see, we can also see that the spread of A green and B red of the data of the individual sites is a bit higher than that of light blue, which has most of the sites close to the center, and of orange as well. And with that, I leave it to you to conduct Manova in the exercise 10, and you should do this for the class data set that it's contained in the library Chemometrics. This data set requires some pre-processing, so we only use certain data of this data set. Try to find out what these functions do in this data set, and then run an ANOVA and also conduct an RDA on the groups and visualize the results. So with that, I thank you for your interest and hope to see you soon with another video.